Okay, so I would like to welcome everybody to our first installment of our new public speaker series called A Deep Look into the Future of Biology. The campus, as you may know, is approaching its 60th anniversary, and in biological sciences we decided to get a jump start on celebrating this anniversary because then we can celebrate for the next 2 years instead of just for 1 year. And then we can also celebrate when we graduated our first students about 4 years later. So this sounded like a great idea to us. And what we decided we wanted to do was invite the community back to UC San Diego to meet some of our fantastic people, our faculty and our students who are really driving scientific discovery forward, and introduce you to them and to the discoveries that they're making so that you can see what we're doing here. Because we're a public research university and we really belong to you, the public. And so we're just really pleased to be able to share with you what we've been doing over the last 60 years and what we've achieved as a division. The campus, of course, is just undergoing remarkable transformations and growth, and we're now a top-ranked public university. And I think you'll see in the talks tonight and in the future 3 events the passion and the creativity that have really driven discovery, not only in the Division of Biological Sciences, but also on the whole campus, and kind of the quirky fun that we like to have with our science and our education as well. So I'd like to just welcome you here, and a special welcome to our alumni that have come back to campus. We're really pleased to be able to welcome you back here to visit the campus and see what we're up to. I hope that you'll all be able to join us for the next 4 events, the next 3 events after this one as well. This event is co-hosted by the Division of Biological Sciences, the Graduate Division, and the Alumni Office. And I'd like to thank the Graduate Division and the Alumni Office for being our great partners in this event, because we wouldn't be able to do this without partnerships across the campus. So in tonight's talk, our speaker series, you will see 4 individuals who share a common passion for their particular organism or method of study. And you'll see how this passion and commitment to their scientific question leads them to really drive the development of new technologies and also the applications of these technologies in ways that can have unexpected sometimes expected benefits on human health and the environment. And so I'd like you to keep in mind that all 4 of these speakers are fantastic scientists committed to fundamental discovery. So not only are they making fundamental discoveries into how life works in their various systems, but also they're keeping their eye out for ways that this might have a future benefit to humanity. So without further ado, one other piece of business. I forgot to say that I'd like to ask the audience to hold their questions until the end, and team members will have cards to hand out where you can write your questions, and then we'll read them to the group after the end of all the talks. And this is because the talks are pretty fast-paced, and we want to keep the flow going and then give you time to answer the questions at the end. Okay. So our first speaker is Professor Stephen Mayfield, who is a distinguished professor and the Director of Food and Fuel for the 21st century. Steve is passionate about algae in all of its various ramifications, and this passion has led him to harness algae in a variety of really creative ways, from, as you'll hear tonight, making flip-flops, surfboards, and nutritional supplements. You might have heard about a study that he was just on that was aired on NPR last night. So he is really passionate about finding ways to use algae to do things that are good for the planet. So please join me in welcoming Steve Mayfield.
I think I'm good. Okay, thanks. We only get 7 minutes, so I'm going to talk fast. Actually, it doesn't matter. I talk fast no matter what. So what I'm going to tell you about today, when I got my PhD, which is amazingly enough 35 years ago, I was like most people who got their PhDs at that time, pretty much basic scientist. I got mine in genetics. I want to understand how genes work and how they function. And I used to tell my mom, yeah, Mom, I work on gene regulators, on promoters. And she would always look at me and say, well, if you're excited about it, honey, I'm excited about it. But what happened over the last 35 years is I kind of noticed that we're trashing the planet. In fact, I would actually say today the planet is on fire. Quite literally, we've seen it in the news just over the last few months down in Australia. This is just one example that I'm going to give you. What I work on could cover a lot of different things, but this is one of them. So our planet is covered in plastic trash. I think we all know this. Just some numbers which are staggering. There are 17 trillion pounds of plastic garbage on this planet. That is 2,600 pounds for every man, woman, and child alive today. There was no plastic when I was born in 1955. It didn't even show up until 1960, and we didn't really get going on it till 1980. We have accumulated that much plastic garbage in the last 40 years. 18 billion pounds of plastic flows into the ocean every year. And I'm sure you guys have seen the pictures of, you know, the whales and the dolphins and the fish washing up dead because they've eaten this stuff. That has got to change. And what I'm going to tell you about today is I think we do have an opportunity to change it. But I want to go back, if I can get this stuff. But that's a symptom. And I want to just briefly discuss what the underlying problem of this. Where does this come from? This is petroleum utilization over the last 10,000 years. And we essentially didn't touch that stuff until 1903. And in 1903, Henry Ford invented the assembly line and learned how to put the Industrial Revolution on steroids. And we are going to burn through 300 million years of accumulation. Who knows where oil comes from? Where does petroleum come from? Melted dinosaurs? Ancient algae? Who knew that? Yes, of course, that's what it is. It's ancient algae. It's 300 million years of algae growing on this planet, settling to the bottom of the oceans and being covered, you know, by soil and soot. And then over time, it converted and became crude oil. All right? But let me show you what, in addition to that, here's the second thing, and that's if we put world population overlaid on top of petroleum utilization, you can kind of see that there's a bit of a correlation there. And it's not to say that those things are cause and effect, they're just interrelated. And how they're interrelated is once we started the Industrial Revolution and then laid on top of that the Green Revolution, those two things combined, we use fossil fuel to make really inexpensive food, and then we transported it all over the world to places where agriculture would not support anybody, but now can because we can transport it, and that allowed 7.6 billion people to be on this planet and to not take very good care of it, unfortunately. So this is the underlying problem, but one of the symptoms is plastic. And what I'm going to tell you about today is maybe we can at least treat that one symptom. All right, so how are we going to treat it? So it turns out that the reason there's so much crude oil underground is because those little organisms, algae, are the most efficient organisms on the planet at converting sunlight and CO2 into products, right? Maybe 20 times more productive, more efficient than terrestrial crops. So the idea is pretty simple. We're going to take sunlight and we're going to take CO2 and we're going to take our little photosynthetic engine of algae and we're going to use that to make different products. Those products could be biofuels, and I'm funded by the Department of Energy to one day make biofuels, but those are very cheap and require a large scale. They could be things like therapeutics, and we occasionally make those in our labs, or they could be plastics, right? And I'm going to show you now about the plastics we make. So the concept is really simple, right? Here's the algae polymer carbon cycle. I'm going to take CO2 from the atmosphere. I'm going to capture that into oils in my algae. Remember I told you that petroleum comes from algae, plastics come from petroleum, therefore plastics could come from algae. So pretty simple concept. I'm going to capture that CO2 inside the algae. I'm going to take that oil, I'm going to convert that to one of the plastic precursors. In this case, we make polyurethanes, and then we're going to make products out of that. And then if we do the chemistry all right, because this came from a biological source, we should be able to turn it back into a biological source. So that's the simple idea. It becomes a little more complex when you actually have to make something, especially a product. That picture in the upper left-hand corner is an algae farm. Actually, it was a company that I started about 10 years ago called Sapphire Energy. They were going to grow up that algae to make biofuels, but instead it got converted to making omega-3 fatty acids. The byproduct of that is another oil, which is actually the one that we use to make our flip-flops. The basic building block of plastics is called a polyol. And so we take that algae oil and we make it into a polyol. And if that polyol is linear, you can make soft foams. And if that polyol has got lots of branches in it, you make rigid foams. First thing we made was rigid foam surfboards. I personally think that's the most important product on the planet. Not everybody agrees with that. But once we made those, then we were quickly approached by many of the shoe companies that said, hey, if you can make rigid foams, you can make soft foams. Work with us to make soft foams. Work with us to make footbeds and midsoles for shoes. So we set about to do that with a couple of big industrial collaborators. That turns out to be rather difficult. Why? Because it turns out that those little pieces of plastic on the bottom of your feet actually have a very demanding specification. What's a specification? A specification is you got to get the density just right. You have to have the tensile strength just right. The tear strength, which actually turns out to be the most important one if you work on flip-flops, you actually have to have 200 or sometimes 300 pounds pressing on those things, and they better bounce back after every step, and you better not tear the straps out. So it took us actually several years to meet specification. But we finally did, and we now have flip-flops that are commercially viable. Importantly, we kept that chemistry such that at the end of their life, they would biodegrade. Here's just a piece of foam that I took home. This is one of our algae foams on the left. I took it home and threw it into the compost pile outside of my house, left it there for 14 days, pulled it out, and sure enough, it was covered with bacteria and fungus. We've now managed to identify what all those are, including the enzymes that come out of them, so now we have completely biodegradable flip-flops. So what did we do? We had to go out and now commercialize these things. Well, it's one thing to have a product, it's one thing to have a foam that biodegrades, but to actually turn it into a product turns out to be a pretty complex set of things you got to do. You actually have to design one. You have to have product development. You got to get supply chain. You have to do all of those things. Super fun to learn those things. Very frustrating because many of them in theory work really well, and in practice they're kind of painful. But we actually got them going, and a year ago March we made our first demos, and I've been wearing those things every day since March, and they are working beautifully. Because of that, we now have a collaboration with Reef. I'm not sure if I can really announce this yet or not. We've gone into production down in Leon, Mexico, and these things will be available on the market in June of this year. So we will actually have a product that you can wear on your feet that when you're done with it, you can throw them in your compost pile and they will biodegrade, okay? All right, so where do we go next? There's lots of places we could go. We get lots of opportunities. We have the most efficient organism on the planet. So now, flip-flops were our first one. I think our next one is going to be primarily animal food or human food. And then eventually we will hit the scale that we could make biofuels and we could fly airplanes and drive cars on them. But by then, Elon Musk will have figured out the electric car and we'll all own one of those. So I'm going to leave it right there. Thank you for your attention and pass it off to our next speaker.
Thank you. Thank you, Steve. So our next speaker is Marty Yanofsky. Marty is a distinguished professor in cell and developmental biology and the holder of the Paul D. Saltman Chair for Biological Sciences Education. He's also the winner of many prestigious scientific awards, and his passion is plants, and specifically how from a single plant seed you're able to generate a plant with all of these different forms, and how the flowers form and how the seeds ripen and how fruit ripens. This, of course, has huge importance because much of our agriculture is based on harvesting seeds and fruits. And Marty's work has applications in agriculture that he'll tell you about today. What he won't tell you about today are his many just spectacularly beautiful papers showing how it is that flowers develop and how fruit ripens in plants. But rest assured, he has made fundamental contributions to discovering how these mechanisms work. Marty received his bachelor's degree from UC San Diego, and so he's a UCSD alum, and he received his PhD from the University of Washington, where I was an undergraduate also. So please welcome Marty Yanofsky.
Hi, everybody. I'd like to start with maybe a serious question, and that is, how is it that we are going to feed everybody on this planet in a sustainable way? And we just saw from Steve that the population is increasing. How are we going to feed all those people in a sustainable way? Now, the answers obviously are complicated, but I want to illustrate something that I think is going to be part of the answer, and that is increasing the yield of crop plants. And just as an example, if you are a farmer and you're growing, let's say corn on 10,000 acres, and if you can double the yield of that corn without increasing the inputs, that means in theory you could get the same amount of corn, the same yield out of that farmland with half as much land, half as much water, half as much chemical pesticides and fertilizers. So increasing the yield of crop plants is going to play a very important role in sustainable agriculture in the future. And I'd like to tell you about one of the projects in our lab that has increased the yield of one of these crop plants. This is a field of canola, and I think you're all familiar with canola. We use it to cook with all the time, and it's marketed around as the healthiest vegetable oil, and it's grown around the world. But what exactly is canola, and where did it come from? Canola was developed in the 1970s, so it's only been around for about 50 years. It was developed by plant breeders in Canada. And importantly, the canola seed is about 50% oil. And you can see this seed pod that's opened up here. You can see these oil-rich seeds here. So 50% oil, that's a lot of oil in every single seed. But one of the problems that farmers face is that a lot of these seeds end up on the ground instead of in the harvesters. And the reason for that is that there is premature seed pod opening. These seed pods, which are kind of like a pea pod, they open up and disperse the seeds onto the ground before the farmers can come through the fields and harvest them. Farmers go to a lot of trouble and do many things to minimize the loss of seed yield. And one of the things they do is this odd technique called windrowing. And you've probably not heard of that unless you're a canola farmer. And what they do is they literally, they have machinery that goes through the field about 2 weeks prior to harvest, and you can see these here, and they literally chop down the canola plants kind of at the ankles there, and they lay them down. They lay them down in the direction that the winds blow. And by doing that and laying them down like this in these windrows, they minimize the loss of seed yield because the canola plants, as you can see over here and here, they brush against one another, causing those pods to open and the seeds fall to the ground. So this, you can imagine, is very expensive and time-consuming, and farmers don't want to have to do that. And so farmers have been asking the plant breeders for decades to find some solution to this premature pod opening. Now, in my laboratory, we've been studying this small laboratory weed, as we call it, called the Arabidopsis. It's the model organism, if you will, for plant biologists around the world, similar to fruit flies for animal research. And importantly, the Arabidopsis plant is in the same family. It's a very close relative of canola, okay? And that suggests that any discoveries that we make in Arabidopsis can easily be translated and transferred to canola, which is a crop plant. So we used Arabidopsis to identify the genes that are involved in fruit opening or seed pod opening. And you can see one of these seed pods here. It looks, again, like I mentioned before, just like a pea pod that you might open at the dinner table. And you can see this narrow seam here that's starting to open here. As that opens, the seeds fall to the ground. And it's actually a very interesting mechanism, and I don't have time to go into the details today, but there's a narrow strip of cells here along the entire length of the fruit, just a couple cells wide that are connected to one another. Those cells secrete a glue that glues this cell to this cell. And then when it's time for the fruit to ripen and for the pod to open, those very same cells secrete an unglue, or something that degrades the glue, so this cell isn't connected to the other cell. They come apart, and then the seam opens, and the seeds can be dispersed onto the ground. So as I mentioned, we used Arabidopsis, this model organism, to identify many of the genes that are involved in opening of the fruit. And once we identified those genes, we realized that it should be relatively straightforward to use that technology to solve the fruit opening in canola. So we then started a collaboration with a seed company, the largest seed company that produces canola seeds around the world. We started as a collaboration, and ultimately, after another 10 years of further research at this company, they started producing a product based on our technology. They call this technology pod guard because it guards the pod from opening. And this is how they market it. Now, it's been a real boon for farmers who grow canola around the world, especially in Canada, where it's grown, it's the largest crop in Canada. So not only do the farmers not lose any of the seeds anymore, they get much higher yields, of course. And I think just as importantly, it enables direct harvesting. And by that, I mean that the farmers don't have to do that windrowing anymore. They can let their crop mature fully and then harvest it and not worry about losing any of it. I think it's probably best illustrated by showing a little video of what those seed pods actually look like. If you take one of those standard seed pods that have been around for the last 50 years and you just touch it, it explodes open and the seeds fall to the ground, and that's the problem. And the ones that we helped modify with our technology don't have that problem. You can see that in this video. So this is just a field, and the farmer is touching the standard variety, and you can see the seeds dispersing. They normally fall to the ground. That happens all the time. And from the same field, with the variety that we help modify, the pod guard technology, it doesn't happen. So it's very dramatic. So I have to say it's been very satisfying. Maybe the most satisfying of all is just reading farmer tweets. Some of the farmers call this game-changing technology because it completely changes the way canola agriculture occurs. So I want to mention the people in my lab that actually did the work. Three outstanding scientists, Sarah Lodrigan, Christina Ferrandis, and Sherry Kempen. And this work would never have seen the light of day without the collaboration and the work done by the scientists at BASF, Benjamin Laga and Bart Lambert. And I really appreciate all of their efforts because it took another 10 years, as they mentioned, for them to do all the field trials and getting it working in canola. So that's what I have to say. Thank you.
Thank you. Okay. Thank you, Marty. So our next speaker is Beverly Naigles. Beverly is a third-year PhD student in the lab of Professor Nan Howe in molecular biology. Nan's lab is interested in studying how the genetic circuitry in cells is wired such that you get the right outputs for stress and during aging. And so he's trying to understand how cells are wired to do the right things at the right time and in the right place, because perturbations in that can cause human diseases such as cancer. Beverly's has a couple of passions. She'll be telling you today about her passion for macrophages and for understanding how the genetic circuitry in these cells is wired to let these cells do the right thing. Macrophages are key cells in the human immune system that they help you survive infections such as the flu and other viral illnesses and bacterial illnesses as well. Beverly received her bachelor's degree from Brown University, and we're happy that she came to join us here for her PhD program. So take it away, Beverly.
So I want you all to think about the last time that you were sick. Probably maybe someone who you knew was sick, and then a couple days later you had a fever, and then maybe the next day you felt a bit nauseous, and then a few days later you started to feel better again. That experience of getting sick and then getting better is what happens when your immune system is fighting this infection that you have. And the same way that your body goes through this response to an infection, cells can also respond to an infection. So when the body's infected, for example, with a virus, there are cells in the body that send out this molecule that tells the other cells, we've been infected, we need to do something. And one of the cells that responds to that is a cell called a macrophage, which is the cell that I study. And when macrophages get these signals, they go through this whole series of events that leads to them becoming cells that are specialized to fight this infection. And they can do this in a couple of ways. So one of these ways is by activating immunity. This is similar to the way vaccines work. Another way is by telling other cells to make inflammation, which is related to when you get a fever. And another way is by crawling around the body, eating up the viruses and the dead cells that are left behind after this infection. Now, even though we all go through generally the same process of getting better after we get sick, You might have had the experience that maybe your roommate or your partner gets better a little faster than you do, and you're like, why can't I be like that? So in the same way that people are different, cells are different. But in biology, we used to study cells as if they were all the same, and we would study them at a population level. But as I've told you, we know that cells are not all the same, and in fact, there might be some that respond more or less strongly than others, or others that respond in a completely different way. In order for us to get a little bit of a better handle on what it means for cells to respond differently to a stimulus, I want us to think about the San Diego Padres. This is a hypothetical plot of the average number of runs that the Padres are making per game across the season. And we can see that when there's a coaching change, overall, the Padres are doing better. But when we break that down by individual Padres player, we're able to see that before the coaching change, some are doing better, some are doing worse. And then after this coaching change, some improve, some get worse. Even ones that were doing the same originally are different. So my question is, are my macrophages like these Padres players? Do they respond differently to stimulus? So the way that I can study this is by using time-lapse imaging, where I put cells under the microscope and can take pictures of them for a long time. And I actually grow them in something called a microfluidic device. And what this lets me do is either give them the we're sick, we need to respond signal, or just we're regular and happy signal. And then I can look at them to see how they respond. And just to give you a sense of scale, this whole thing fits in here, which is smaller than a penny. So microfluidics really are pretty small. Okay, so when I do this, what do I see? What you're looking at here are still frames of a video of these macrophages responding to the stimulus. And each of these is a cell, and the red is marking the nucleus of the cell. And we can see that at the beginning, they all look pretty similar. They're all pretty small. But then by the end, and I'm going to zoom in so that you can see this a little better, you can see that some of them still look similar, but this one looks big and it has all this cytoplasm around its nucleus. And so this one, for example, might be ready to crawl around the body and try to eat up some of the things that are happening because of this infection. So I see that, like the Padres, my macrophages are responding heterogeneously. So now the question is, in baseball, if we know these players are responding differently, how do we figure out why? And one of the ways that we do this in baseball is by all of these baseball statistics that we have. So underneath runs, you need to have at-bats, and you need to have hits, and you need to have all these things. And so we do the same thing in biology, and I can do this by lighting up different genes. So the way that I do this is I can actually cut the genome, and then I can put in a fluorescent protein attached to a certain gene. And so anytime the gene is turned on, my cell will light up. And I can do this for multiple genes in the same cell, so I can see multiple things lighting up in the same cell and track different statistics in the same cell. And then I can look at this cell and say, what did it do? Did it create inflammation? Did it activate immunity? Did it crawl around eating things? Did it not do anything? Okay, and this is all really cool, but what does this have to say about biology? Well, when you have, we're going to just take as an example an inflammatory fever response. If every single cell responds to this signal and creates this fever, you're going to have much too much fever, and you're going to be extremely sick because of your own immune system. On the other side, if none of the cells respond to this virus, you're not going to have any fever at all, which means that the virus is going to be perfectly happy and keep hurting you itself. What we get when we have this heterogeneous level of response is an intermediate amount of fever, which is just enough to help kill the virus without killing all of our own cells. And in my work, understanding the processes that help us get to this heterogeneity, as well as the different choices that the cells are making, can help us modulate the immune system to make sure that we can always get to this Goldilocks place. And I want to also tell you that this is just one example of the ways in which this cellular heterogeneity and diversity is important for biology, and I'm really excited about the way that us and other biologists are starting to think about this more. I also just want to thank, as Kit said, I'm a graduate student, so I work in Nan Howe's lab, so to thank my advisor and my committee and all of my lab members for all of their help, and thank you so much.
Thank you. So our next speaker is Elizabeth Villa. Elizabeth is an assistant professor of molecular biology, and her passion is developing new methods to snoop inside cells to see the inner machinery of the cells and to look even deeper inside the cells. And she will tell you about how she's done that here tonight. And Elizabeth did her PhD in biophysics at the University of Illinois Champaign-Urbana, and then she did her postdoc at the Max Planck Institute. Please welcome Elizabeth to the stage.
When you think of a disease like Parkinson's, typically you think of an entire brain or a region of the brain that has been affected, and normally you'll be presented with images like this. If you're lucky like me and have a friend like President Madigan, an assistant professor in the division, you'll get a Christmas cookie that looks like that. It goes the same with viruses. Typically with the coronaviruses, we're very excited when we get the first images of them, but that doesn't really tell you how entirely at the molecular level these diseases operate inside your body. What really happens, everything that happens inside your body is performed by molecular complexes or biomolecules that are made of chemical entities and come together to form these machines that perform all functions in your cells. This is how we study the structures of these proteins. We isolate them biochemically and look at their exquisite, beautiful structures. But biomolecules, of course, don't exist in isolations inside cells. They come together, they're very social, they have friends. Every given molecule interacts with many others to form networks that are really exquisitely controlled inside cells that affect their function and things like that. These are microtubules. These are highways inside cells where everything gets transported with one side of the other. Imagine if we could build and unbuild all of our highways every day as we need them, just exactly when we are going to go and walk to bring one thing from one side to the other. And of course, all of this control that works incredibly well sometimes gets affected in disease. And so my drive had always been, why study these molecules and their mechanisms in isolation? Can we find ways to look at them inside cells, something exactly like this, but with real data as opposed to beautiful animations? Importantly, some molecules are difficult to study even in isolation. Notably, there's a protein called LRRK2, which represents most cases of genetic Parkinson's disease. And this protein was so difficult to characterize that a few years ago, the Michael J. Fox Foundation sent protein to space to see if they could form crystals under zero gravity conditions. And it didn't. So the structure of this protein remained elusive, and a lot of money went into that. And so there's a lot of interest in looking at how the molecule affects the cells in disease, but also how it works and what this structure is. In order to take onto this new challenge, I went to Munich to do my postdoc, and we started developing these technologies that my lab continues to develop today, in which we either adapt technologies that exist for other areas, or just combine together technologies and build new ones. So around cryo-electron microscopy, we've built a lot of technologies to be able to literally open the hood and look inside the cell at frozen molecules caught in the act of interacting with each other in different places in the cell. And we do that by combining fluorescence microscopy, just in the way that Beverly showed you before. And we developed a technology called cryofib milling that I'll show you right now. And eventually we take 3D maps of entire cells. So this is a movie that illustrates the microscope that we co-developed with a company. And as you can see, the chamber has two beams. You'll see this in a moment. We have an electron beam, which is the one that you take pictures of, like fly eyes or bed bugs in your doctor's office, and an ion beam that's used in material sciences to edit chips or micromachine materials. We adapted it so that we could put frozen cells inside it and it's really like a lightsaber, and we blast away parts of the cell, and we leave in the middle a window that contains the molecules that we're interested in looking at, and we call that a window into the cell. We have to move from that microscope to a different microscope, which is a very powerful transmission electron microscope, or TEM. And in that, we basically take a CAT scan of this region of the cell. And it's exactly the same, except our samples are 1,000,000 times smaller than you. They're better behaved than you inside the CAT scan. And we take pictures in different orientations, and we combine them together to create 3D structures of cells. This is data from my lab. And you can see I'm going up and down in the screen, and it's in black and white. But you can see, for those of you who remember biology, the nucleus is on this side of the cell. In green, I'm showing you mitochondria. These are similar to the molecular networks that I showed you in green and red. And these are ribosomes that make cells. These cells have not been altered in any way. They have no artifacts from the preparation. We don't need any cell preparation. We could take some flakes of your skin, grow them on a grid, and look at this type of behavior nowadays. So of course, as you can imagine, we're very excited about this technology, and we've been applying it to a lot of different areas. Parkinson's disease just happened to have gotten hold of my heart recently. We can grow cells, these are human cells, that have a mutant LRRK2, which has been fluorescently tagged with a fluorescent protein. In this case, it looks like a jellyfish. So we know where these molecular networks that may lead to pathology exist. We can zoom into these molecular networks, make one of these windows into the cell with a lightsaber, you see it over there, and then take it to the other microscope, and then zoom in. And what we see is that our protein, LRRK2, is labeled in green, and it's co-localizing with the highways that I told you about before. And these highways are in charge of transporting everything in cells, including neurons. So we were curious to see what was happening over there. So if we take a look at one of these areas where we have these block highways, where you now see is this is, again, one of these tomograms. And these little sticks that you see over here are the highways, and they're completely shrouded with this protein LRRK2 that binds to them, it seems, irreversibly and seems to block their way. We can zoom in farther into these structures. Now the highway or microtubule here, you're going to see it in white. In a minute. Right, in white. And the LRRK2 structure from two helices really wraps around it completely. We can zoom in even farther, and now we can determine the atomic structure of this protein for the first time. So where space failed, we prevail. We're able to map all of the mutations. This is a really interesting protein. We just started scratching the surface, but we're very excited to be able to contribute to this field. And even happier to know that at UC San Diego, there's a number of investigators interested in Parkinson's, and we're slowly coming together. For example, Andreas and Susan are collaborators for this project. Andy Shao, together with Sam and Andreas, we're also looking at inhibitors that we think we eventually will be able to put into the market. Annie looks at entire brains of patients at the molecular level as well. So we're all trying to come together to study this disease. And so it's a very exciting time. And this is Cressida, who also studies Parkinson and makes cookies. Of course, we've been applying this technology to many other areas. My dream is, this is a beautiful painting for David Goodsell, who's a local scientist and artist. And once I sent this picture to David, where this is superimposed to some of our data, so you can see the match is amazing. And so our goal is to use artificial intelligence and other technologies to be able to mine this data to understand, for example, how chromatin works in disease and in health, and really to put together these molecular networks and understand how they're assembled to be able to understand how you're made, which is, of course, it goes well beyond the linear sequence of DNA in your genome. And so with that, I'll show you the people who actually do the work. And thank you very much for your attention.
Right, thank you. Okay, let's give a big round of applause for our 4 speakers. Thank you. Okay, and we will have time for some questions. So I'd like to invite the speakers up, and maybe we should move the chairs a little bit closer to the front. Okay, that's great. Well, we could stand. Do you guys want to stand or do you want to sit? You choose. Speaker choice. Okay, we can stand. Okay, great. Perfect. Okay, so, we have cards. So if you could write your questions down and pass them over. I'm going to get us started with just a few questions. I'd like to start with Beverly. You have a very special activity outside the university. I'd like to invite you to say a few things about that.
So I spend a lot of time in jail. I run a science outreach program with the grad students here in the San Diego County reentry facilities, where we teach science in a really accessible, hands-on way to groups of adults who are incarcerated. This program is really great for everybody. I think that our students really enjoy getting to learn about science, and they get to ask us all sorts of questions about things. We got asked last week why mustard helps treat heartburn. So things like this, and I think that they really enjoy it, and it really helps them have a more positive sense of what is science and realize that anybody can be a scientist. You don't need to have a PhD. And then we grad students also learn a lot from our students who are incarcerated. And so I really enjoy it. It's really meaningful work for me. And it's a lot of fun.
That's great. Is this working? Yeah? Okay, so thank you. No.
Is that working?
Oh, I will. Okay, so my next question is for Steve. Steve, when we come back in 10 years to celebrate the 70th anniversary, what will algae be doing for us and the planet?
Well, first of all, we'll all be wearing algae flip-flops at that point. We will have algae sneakers. But honestly, I think polymers are important and it's a good way to get started. But I think what's quite clear to me is that we have to change the way we do agriculture. Right now, because the rest of the world is coming up to our standard of living. And as you rise up in your standing of living, you tend to eat more animal protein. So it's for that reason that we are deforesting the Amazon and big parts of Southeast Asia so that we can grow soybeans and corn and feed those to cows so that we can eat them. And that has to change. And I don't know how many of you know what Impossible Burgers are, but Impossible Burgers are the new fake hamburgers that are coming out. And I actually believe in 10 years' time, there will be no animal protein sold anymore. That we are getting so good about making synthetic proteins that we can then texture to feel like it. So plant-based proteins that we can texture to become food. So I'm on my way to being a vegan, but I'm not on my way to not eating protein. We will eat lots of that, and I think all of us in this room will as well.
Thank you. Marty, you were an undergraduate here, a few in the 70s, is that right? Could you tell us a little bit about the changes you've seen and what remains the same on the campus?
Yeah, I started here as an undergrad in 1974, and I think there were a little over 7,000 students, and we're up to about 30,000 now, I think. We're going to 40,000. So yeah, it was a much smaller place. I think the thing that I remember the most is that there were two places to eat on campus. There was the Ravel cafeteria, which is right over here, and Muir cafeteria, and the food was lousy. So that's what's changed probably the most. But I have to say that for me, looking back there in terms of my career opportunities, it was 1974. I got a job as a dishwasher in a laboratory, and that was the best thing that ever happened to me because that led to doing research, and it was just an amazing opportunity. And while I was a student here for 4 years, I had this dream of someday coming back as a professor, so I'm definitely living my dream.
And we're glad to have you, so thank you. Okay, I have some audience questions for Elizabeth. Are the changes in the macromolecular structures after freezing of cells the same in all cell types, or do they vary?
Good question. To the best of our knowledge, they're the same, which means we freeze cells to liquid nitrogen temperatures so quickly that water doesn't have time to form crystals. So the molecular complexes, as far as we can see them, remain exactly in the same conformation as they would when the cells are alive. In fact, a pet peeve of my lab sometimes is to bring the cells back to life. I mean, this is how we freeze cells to store them. So with bacterial cells and sometimes with mammalian cells, a large population of them will just come back. Not after we lightsaber them and nuke them in the microscope, but before that. So the structures are very well preserved, and so to our knowledge, this is the most exquisite preservation that exists in terms of that. What we see in this data is literally the conformation of the proteins as they exist when they're moving. And in terms of cell size, we're limited sometimes by the size. We're moving towards being able to freeze better so that we can do tissue directly, plants, and other things. So there's a lot of work yet to be done.
Thank you. Is there any, can you, can the samples be thawed, and then can you do interesting things after you thaw them?
I think I'm more interested in freezing them at a very particular time. So we're trying to build microfluidic devices to freeze cells while we're doing live cell imaging, so we'll be one and the same at some point. I don't know if I would trust the thawing process in terms of studying a cell as if it was a natural living cell, but they certainly, after a couple of cell divisions, they seem fine.
Okay, we have an audience question for Marty. Do farmers find the pod guard seeds hard to open or harvest? Is there any loss of efficiency there?
So the answer is no, they don't have any problems opening them. And the reason is because researchers who developed this, not only starting with us, obviously, but the company that we collaborated with, found exactly the right of pod strength, if you will. And that goes back to the gene that we discovered that's involved in this process. And so we can make those pods, we can glue them shut so the farmers wouldn't be very happy and you couldn't get the seeds out at all. In fact, they would have to develop new technology to open the pods. But it turns out that if you identify just the right allele, as we refer to it as, of this gene, then it has sort of a moderate characteristic or phenotype. And so it doesn't affect the harvesting process in terms of getting the seeds out of the fruit, so we don't have that problem at all. Yeah.
Thank you. Are there any plans for canola flip-flops? Are you two going to collaborate?
So we can actually we could make canola flip-flops, but one of the reasons we use algae, even though you can get many of the precursors from biological sources, we definitely don't want to take food off the table to make something else out of it. Like the fact that we take 40% of our corn crop today, 40%, and turn it into 14 billion gallons of ethanol that we put into our gas tank is not really a sustainable way to run the planet. So as much as we could do it, and I will make Marty a pair of canola flip-flops, in general, I don't think we want to commercialize those.
That's great. Thank you very much. Okay. This has been really fun. I just want to thank you all for being the brave first speakers at our first public speaking event. And I really wanted to thank the audience for coming here this evening and spending a little bit of time with us. I hope that you'll each take away something, a little piece of the passion and enthusiasm and creativity that really drives our science here in biological sciences. And I'd like to invite you to join us at the 3 coming events, which will focus on topics ranging from how the brain works to how new genetic technology is driving and ethics are working together to help cure the planet of various diseases and improve agriculture. And on Earth Day, we'll have a special conversation about the planet and ecosystems and just the natural world around us. So thank you all for coming and joining us, and I look forward to seeing you at future events. Thank you.